Hmm. Okay, before we begin, are there any questions? I have done some grading. My goal is to finish lab worksheet 3 by tonight, and then you can turn it in by this Saturday for the second grading. For lab 4, I'm not sure when I'm going to get it graded, but it won't be very early, so I'm going to postpone. Why don't I do that right now? I'm going to postpone when the second grading will be, and probably shouldn't be here. And so it will not be due this for the second grading this Saturday. It'll be due the next Saturday. Any question about that? I'm going to change the due date for the second grading now. Somebody asked me about when I'll get the second grading done. I've told you before that the second grading will get done whenever I have time. At the moment, I'm struggling to get the first grading done, and the first grading is going to get priority. So the second grading is not going to be graded anytime soon. When it'll be graded, I don't know, but I'll let you know. All right. So the second grading for lab 4, I'm hoping will be February 10th. Obviously, I have to get the first grading done before I can require you to turn in the second grading. Any questions about any of that? All right, let me close that down. Are there any questions at all about the grading? All right, if not, let's go on to the syllabus. And I'm a little bit confused as to where we are. Today is January 30th, and we are supposed to be starting chapter 4, prokaryotic and eukaryotic cells. Where did we finish last time? Did we finish here last time? Where I talked about the gram stain?
Yes.
All right, so we're behind schedule. That's what I needed to know. All right, so this slide showing you the different colors that the cells get in the gram stain. Initially, both gram-positive and gram-negative cells stain blue-purple from the crystal violet. They then stain purple-purple from the iodine added to the crystal violet. The gram-negatives are clear after the decolorizing step, but the gram-positive cells are purple. And then after the safranin step, the gram-negative cells are pink and the gram-positive cells are purple. Any questions about that? All right, let's go into the gram stain and grader procedure. You flood a slide that has a bacterial smear on it with crystal violet for 1 minute. The crystal violet sticks to the peptidoglycan in the bacterial cell wall. Peptidoglycan is considered the primary stain since it's the first dye used, and it colors all the cells the same. So both the gram negative and the gram positive will be stained purple. And yes, it's a bluish purple color. We then stain the slides with iodine. Iodine is not a stain. It is a mordant. It increases the affinity of the crystal violet for the specimen. Iodine actually chemically combines with crystal violet, making a larger chemical. You flood the slide for 1 minute, dump the iodine off, wash with gently with cold water, and the both the gram positive and the gram negative cells will be the same. It'll be a purple color. You then apply the decolorizing agent. The decolorizing agent that I talked about was acetone ethanol. It's the decolorizing agent that we use in the microbiology lab at Clark. It is true there can be other decolorizing agents. There can be acid alcohol, and there can also be just alcohol. All three of these are decolorizing agents. The only difference is the alcohol, you apply it for longer than you do for the other decolorizing agents, because the alcohol isn't quite as good at decolorizing the cells as the acetone alcohol or the acid alcohol. The decolorizing agent washes out the crystal violet from the gram-negative cells, but not from the gram-positive cell walls. They are thicker in their peptidoglycan layer, and so they are able to retain the crystal violet as long as you apply the decolorizing agent for the correct amount of time. If you do apply the decolorizing agent for too long, you can wash the crystal violet iodine out of the gram-positive cells. The decolorizing agent step or whatever is considered the differential step in the gram stain, because the gram-negative cells and the gram-positive cells will first stain differently after the decolorizing step. You then wash immediately the slide with cool water to stop the traces of the decolorizing agent from continuing to work, because they will continue to work on the gram-positive cells, and if given enough time, the crystal violet iodine will be pulled out of the gram-positive cells. So after applying the decolorizing agent, the gram-positive cells will be purple from the crystal violet iodine complex retained in their cell walls, and the gram-negative cell walls will be colorless as they lose the crystal violet iodine complex. Safranin is then applied. It's a pink dye. It does stain all the bacteria cells. It's called the counter stain because it has a different color than the primary stain. You apply the safranin for 1 to 2 minutes. Generally, 1 minute is sufficient for the concentration of the safranin used at Clark College. I've only once been in a lab where the safranin was fairly dilute and you had to apply it for 2 minutes. After applying the safranin, the gram-positive cells will be purple. The gram-negative cells will be pink. And did I talk about why can we not see the pink color in the gram-positive cells? As I state up here, safranin does stain all the bacteria. Did I talk about that? Anyone?
I don't remember you talking about that.
Okay, safranin does get in the gram-positive cells, but when our human eye sees the color, our human eye does not see the pink because it is overshadowed by the purple of the crystal violet iodine complex. So the pink is there, our human eye just does not see it. And that's the property of our eyes. It is overshadowed by the crystal violet iodine complex, at least with the light microscope. If we were to look at the cell with the electron microscope, so let's say this box here is the cell, we would see areas that are purple, like this area here and this area here, and then areas that are pink, like this area here and I don't know, this area here. But that's because the electron microscope blows up the cell much bigger. We can then distinguish the pink areas from the purple. But when we're looking at the cell, we do not notice the pink, even though it is there in the gram-positive cells. And it's just that purple overshadows the pink. It's when not the dominant color. You know what I'm trying to say. It's the color that we will see. Because it is a more intense color. You then blot the slide dry. Let me get some paper. And you blot it with bibulous paper. It's a really thick, almost like a cardboard. And then put the slide inside. That's a little too big for this. Let's make this the slide. And then just tap it down. That's blotting it dry. I don't know if you guys can see this. I'm trying to demonstrate, and it occurred to me you guys probably aren't seeing this. So this is the bibulous paper. You rip it off and then tap it. This is the slide. Tap it down. That's to get the water off the slide. You do not take the slide and move it across the bibulous paper, or vice versa, the bibulous paper across the slide. That will pull off the cells. All you do is you tap it down. Any questions about that? And that's what we mean by blot it dry. That will remove enough of the water that you can even put on oil and view the slide on the microscope with oil. Any questions about the gram stain procedure? All right, let's talk about the basis of the gram staining. The gram-positive cells are purple after the procedure. The gram-negative cells are pink. And why? What's the differential staining? Where does it come from? It's related to the differences in the cell wall structure. The gram-positive bacteria have thick peptidoglycan in their cell wall. The gram-negative have a thinner peptidoglycan layer in their cell walls. The gram-negative cells also have a lipopolysaccharide layer outside the peptidoglycan. It's called the outer layer of the cell wall, or the outer membrane of the cell wall. So the gram-positive cells have a cell wall looking like this. The yellow being the cell membrane, the purple being the cell wall, and essentially it's layer upon layer of peptidoglycan. There are a few other molecules, but we'll talk about them later. Not now. And this is only showing you 3 layers of peptidoglycan. In reality, it's something like 30 to 100 layers of peptidoglycan. The gram-negative cell wall has the cell membrane, a thin layer of peptidoglycan in purple, and then the outer membrane. It is a lipid bilayer. We'll talk more about that later. On the outside of the peptidoglycan, it is part of the cell wall. Let me see if I can blow that up a little. This is showing you only one layer of peptidoglycan. In reality, it's at least 3 layers of peptidoglycan and up to 20 or 30 layers of peptidoglycan. So the crystal violet iodine complex forms in all cells, both gram-positive and gram-negative. The crystal violet iodine complex is larger than crystal violet or iodine alone. During the alcohol acetone decolorizing step, the gram-negative cells have the crystal violet iodine wash out of the thin peptidoglycan layer. The lipopolysaccharide layer is actually disrupted by the alcohol wash, and the lipopolysaccharide layer washes away. The cells, therefore, are colorless as the crystal violet iodine comes out of the cells. The gram-positive cells, the crystal violet iodine complex cannot be washed out of the thick peptidoglycan layer, so the cells remain purple in color. And there you see the gram stain showing gram-positive rods, gram-positive cocci, gram-negative rod right there, gram-negative spirilla right there. And here's some more gram-positive cocci. Any question about that? That's a vibrio. A vibrio is half of a spiral, or you can call it a bent rod. There's another vibrio right there. When you're doing the gram stain, there are sometimes problems. Like right here, how do you read that slide? Where do you read the slide? Do you read it here, here, or there? And is it gram positive or gram negative? There are bad regions in a slide. You ignore the bad regions when you are interpreting your gram stain result. And you look at the good regions. The good regions have individual cells, a layer thick or close to it. And you avoid where there's big clumps. And that's not even a clump. Here, here, and there. This is actually a crystal of crystal violet. Does that make sense? What happens is the crystal violet can be in solution and then come out of solution, forming crystals in the bottle. And then when you apply the crystal violet, especially if you're unlucky, you will get crystal violet crystals on your slide. And you can notice that around the crystal violet crystals, the cells are darker staining. I'm trying to blow that up to show it. Right there, that cell is darker than these cells. These cells here are darker than these cells out here. On this slide is not so much of a problem because these are gram-positive cells. But when you're looking at gram-negative cells, it's frequent that the good areas will be gram-negative, and then the bad areas will be a mixture of gram-negative in this region, and then right here, they'll be kind of staining gram-positive, meaning they'll be staining purple. And that's because the crystal violet from this crystal violet crystal can come out of the crystal and then stain the cells a purple color if they're near the crystal. And the important part is you avoid the bad regions and only make your determination near the good regions. Any question about that? The gram stain doesn't always give consistent results. To get the most consistent results, the gram stain should only be applied on young growing bacteria. That is bacteria that is less than 2 days old, 2 days or less old. I guess less than or equal to is what I meant to say. The gram stain is also not universally applicable. Some bacterial cells stain poorly, if at all. That's mostly the mycobacteria. They have a lot of mycolic acid in their cell wall. The mycolic acid does not readily take up water-soluble molecules. The crystal violet and the safranin are water-soluble molecules in the dye. And so the mycobacterium stain lighter than most other cells. Whenever I've stained the mycobacteria, I did get them to stain. They stained lighter, but they did stain. But some people have told me that they don't stain. And I'm not sure why they got different results than I did. It might be they didn't leave the crystal violet on for a full 1 minute. It might be that they were staining older cells. And the results are not always as clear-cut as the theory suggests. This will become evident during your unknown project, where you'll be pulling out your hair saying, gosh, do I have gram-positive cells or gram-negative? Now, the good news is that you guys will be doing the unknown project online. So you will not be doing the the gram stain yourself. It'll be a stain that I've made, and then I'll send you pictures. Then let me state that if you do use old cells, the gram stain may not give you the correct result. And that's because the old cells have dead and dying cells, and their cell wall will be damaged, and they may not stain correctly with the gram stain. Any questions about any of that? All right, the structure of the gram-positive and gram-negative cell walls not only affects the gram stain, it also affects how the cells are related to different antibiotics. Like the gram-positive bacteria can be killed easily with penicillin G, as well as cephalosporin and many detergents, whereas the gram-negative bacteria are resistant to these antimicrobials. With penicillin G, it cannot penetrate the lipopolysaccharide layer of the cell wall. And if penicillin G cannot get inside the cell, it cannot damage the cell. And so the gram-negative bacteria are not affected or controlled by penicillin G. And that's the same thing with the cephalosporin. The detergents is mostly that the detergent cannot get in the gram-negative cell, that the outer membrane of the cell wall can prevent the detergent from getting in the cell and then damaging the cell. I want to briefly talk about special stains. You're not going to be examined on this, but there is one special stain you need to know, and that's the capsule stain, because some of you will be performing the capsule stain for your unknown project. The capsule stain stains the background of the cells, meaning the slide, and the cell. It does not stain the capsule And so the capsule looks clear, clear zone around the cell. This is a capsule stain using probably two ink dyes, one staining the background, one staining the cell. The next slide, let me go to it, is India ink used as a negative stain. India ink stains the background darkly, and it does not stain the capsule. It does lightly stain the cell a gray color. Anyway, special stains can be useful for identifying capsules. Any question about any of that? All right, if not, that's it for chapter 3. Any question about chapter 3? If not, let's move on to chapter 4. Chapter 4 is the functional anatomy of prokaryotic and eukaryotic cells. Know the terms of this chapter. You should also know how gram-positive and gram-negative cell walls differ. We're going to look at them and talk about the different molecules in the cells. And then lastly, describe the importance of taxis in bacteria. When we're talking about prokaryotes and eukaryotes, you should know that the words are Greek. For prokaryote means pre-nucleus. Literally, pro means pre, karyotes means kernel, but by kernel they're meaning nucleus. And eukaryotes literally translates to true kernel, which we loosely translate as true nucleus. The eukaryotes have a nucleus, the prokaryotes do not. The prokaryotes generally have one circular chromosome. It's obviously not in a nucleus. By a circular chromosome, we mean the starting position here ends up where the ending position is here, meaning the starting position is the same spot as the ending spot because the chromosome is in a circle. The eukaryotes have linear chromosomes, like the X chromosome shown here, where there's a beginning spot on the X chromosome and there's an ending spot on the X chromosome, and the two are not the same spot. The linear chromosomes are in the nucleus. I guess one other difference, which I mentioned but did not specifically state, and that is prokaryotes tend to have one chromosome. Eukaryotes tend to have several to many linear chromosomes, and their chromosomes are in the nucleus. There is no DNA associated with histones in prokaryotes. I should state in bacteria, because bacteria do not have histones. The eukaryotes have their DNA wrapped around histones. Histones are large proteins, and the DNA, let's make that the histone, the DNA actually wraps around it. The prokaryotes have no membrane-bound organelles. The eukaryotes do have membrane-bound organelles. By membrane-bound organelle, I mean an organelle with a membrane around it, such as the Golgi apparatus, or Golgi complex, the mitochondria, chloroplasts, vacuoles. There is peptidoglycan in the cell walls of bacteria, and only in bacteria, not in archaea. There is no peptidoglycan in the cell walls of eukaryotes. And of course, animal cells don't have a cell wall. Prokaryotes divide mostly by binary fission. Eukaryotes divide by mitosis. So let's talk about the cell shape and arrangement of prokaryotes. We'll talk about the cell structures we find in prokaryote cells, and then we'll talk about the movement of material across a membrane, such as the cell membrane. In eukaryotes, we probably will not talk about any of the material in this what do you call it, PowerPoint, because you should have had eukaryotes in your preliminary biology class, your prerequisite for microbiology. So I will not talk about it, except for I will talk about the endosymbiotic theory. It's a theory to explain how eukaryotes obtain their mitochondria and their chloroplasts. So prokaryote cell size and shapes, the average size ranges between 1 and 10 micrometers. Most prokaryotes are between 1 and 2 micrometers. The basic shapes of prokaryotes is they have round cells that we call a coccus, shown here. They have rods that we call bacillus, and you may use the term rod as a scientific term to name bacillus, because these two are used interchangeably. And then we have spirals, shown here. These are the basic shapes of most prokaryote cells, especially the bacteria. There are a few other shapes, like there are spirals, which is a bent rod, or a vibrio, which is a bent rod, or you can call it a half of a spiral. So if this were to bend right there, that would be a spiral. The spirals are spirillums and spirochetes. The spirochete has many more, waves to the cell than the spirillum, which is essentially an S shape. There's a little difference in the cells, and that is the spirillum is a rigid cell that can take a corkscrew shape and a corkscrew movement. We'll talk about that later. The spirochete is more flexible, and it has multiple bends. It can also bend the cell. Let me see if I can demonstrate this. So let's pretend this is the spirochete. It can actually bend on itself like that, so that the tips can touch each other. Like I said, the spirochete is more flexible than the spirillum. You have some unusual prokaryote shapes, like there's a star-shaped Stella bacteria, that's a genus. And the square, actually, I think it's not square, but rectangular Haloarcula. This is an archaea bacteria. And there's the actual electron microscopic image, or there's the electron microscopic image for these. This is an artist's rendition. Let me talk about one other shape. We'll come into it here. And I don't know why it's even down here, because this is where I'm talking about the cellular arrangement, also called the cellular growth arrangement, where the cells can stick together and then form a cellular arrangement. Coccobacillus is not a cellular arrangement, it is a shape. And what it is a shape in between a bacillus and a coccus. If you have a rounded bacillus, meaning the edges around it, and then you get the cell smaller and smaller and smaller, you'll eventually get a coccobacillus, a shape that's similar to a coccus, but it's a little bit elongated, so it's called a coccobacillus. Any question about any of that? Most bacteria have a single shape, like the Haloarcula. All of them are rectangles. And the Stella, even though they do have slightly different shapes, they are all star-shaped. 5 stars, 5 edges, whatever you want to call it. And then the Vibrios, they're all bent rods. The Spirillums, they're all spirochetes. The point is they all have the same shape. So most species are monomorphic where they have a single shape, but there are a few bacteria species and a few archaea that are actually pleomorphic. And this is where the species takes on many different shapes, as shown here. Let me blow this up a little bit. Here you got a dumbbell shape. I'm not sure what you would call that shape. Teardrop shape. That's close to a cockeye, that one there. And that's close to a coccobacillus. That's sort of a bent rod. There's a better rod right there. And then that one's just an odd shape. And that one's a real odd shape. Pleomorphic. Many of the pleomorphic species are bacteria that lack a cell wall. And that's why they're pleomorphic, because the cell shape will depend on what shape the cytoplasm takes, and the cytoplasm can take more than one shape. And so, like I said, many of the pleomorphic bacteria are bacteria that lack a cell wall. I'm not sure if there's any species of a pleomorphic bacteria that has a cell wall. I have to look that up, I guess. Usually when we're talking about pleomorphic, we're talking about the shape of the cell in any one given environment. It does not usually mean the shape of the cell in different environments. There is one species that I know of that takes on a different shape depending on what environment it is in. Now, let me see if I can remember. I think it's a rod, like when it's grown on a solid dry media. And then when it's in a liquid culture, it's a spirillum. I think that's the species I'm thinking of. And usually when we're talking about pleomorphic, we're talking about the shape of the cell in one environment. Most bacteria do not change their shape in different environments, but I know there's one species that does. And there's probably others I just don't know about. All right, let's talk about the prokaryote cellular arrangement, also called the cell growth arrangement. These are what arrangement the cell forms if the cells divide and then the cells stick together. The cocci can have many different cellular growth arrangements. You can see singlets, that's a single cocci, no cells attached. You can have diplococci, that's 2 cocci attached to each other. You can have streptococci, and that's a row of cocci all lined up in a single layer. There's one ring of streptococci here. This is a diplococci there. The tetrads are hard to see here because they have so many cells. There's cells on top of each other, but there's a tetrad right here. 1, 2, 3, 4. 4 cells stuck together is called the tetrad. You can have Sarcsinia. This is 8 cells stuck together. Very hard to see them here. For one thing, even here you only see 7. The 8th one is right here, but it's being blocked by these 3 cells, so you can't see the 8th cell. And right there, you can see 4 cells of the Sarcsinia, but maybe that's a 5th cell. You can't see the rest of the cell because they're blocked by this one here. And this is another picture where there's too many cells on top of each other, so it's very hard to see the cells because this is one Sarcsinia here, this is one here, and this is probably one there, and maybe even one there. And they're just too many cells on top of each other. You can also have cells in a clump, and that we call a, see if that goes away, that we call staphylococci. These are cocci grown in a clump. And staphylococci actually means cocci in the shape of a grape cluster. So that's what staphylococci means. And if you put a stem right here, and then this would be the point of the grapes, that would be the grape cluster. Trying to think, are there any other that I need to talk about? I don't think so. The bacilli have cellular growth arrangements, but they have fewer than the cocci. You can have a single bacillus, shown here. You can have diplobacilli, shown here. I don't see any diplo here, but there's a singlet. You can have streptobacilli. That's the bacilli that are hooked on the tips this way. And so that sort of looks like a string of sausages. If any of you have seen that, especially in an older movie. I think occasionally I can find hot dogs in a streptobacilli pattern as well, but not often. And there you can see one cocci, another, a third, maybe even a fourth in the streptobacilli. Excuse me, not cocci, bacilli. And here we have Coccobacillus. This is a real picture. And you can see that this is more elongated. Come on, mouse. More elongated than a cocci. That looks like a cocci there, but even that one's elongated. But not quite elongated enough to be a bacillus. And that is not a growth arrangement. This is a cell shape. The other cell shape is when the bacilli are not connected on the tips, but on the long edge. And this we call a palisade. And that's because if you flip that lengthwise, let me see if I can end the show and call up paint. This used to be a very common fence, and that is they just put one board here, one board here, one board here. And then this will be the soil here. If you read Mark Twain, I think it was Tom Sawyer was having to paint a palisade fence. So the palisade fence was mentioned in Mark Twain. And if I remember correctly, Tom Sawyer was trying to convince Huckleberry Finn how fun it was to paint the fence. And so he got Huckleberry Finn to start doing his work, Tom Sawyer's work for him, meaning Huckleberry Finn ended up painting the fence for Tom Sawyer. Any question about any of that? You should note that the word bacillus has two meanings. A bacillus, if it has a lowercase b, is the shape of a bacteria. It's a rod. But a bacillus with a capital B means that this is a family of bacteria that is called bacillus. And also don't get confused with the word streptococcus, which is a family of bacteria from the streptococci, which is the growth arrangement of cocci. Okay, any question about any of that? All right, so here are some typical structures we find in a prokaryote cell. The red structures are seen in all cells, or at least all bacteria cells, I should say. The other structures shown in black are structures that you can find in bacteria, but not all bacteria have all structures. Like only some have a pilus, only some have a capsule. Most bacteria do have a cell wall, but not all bacteria have a cell wall. The structures found in red are found in all bacteria. That would be the cytoplasm, the ribosomes, the plasma membrane, and then the nucleoid region, and that contains the DNA. There is no separation between the nucleoid region, where the DNA is floating, and the cytoplasm, because the DNA is actually floating within the cytoplasm. That region does stain differently where there's DNA as opposed to where there's not DNA. But there's no boundary between the two. And it's wherever the DNA will move when it's floating, that's the nucleoid region. Okay? Any question about any of that? Let me state that the cytoplasm, ribosomes, and plasma membrane are found in all cells, although usually people don't say the ribosomes are, because the ribosomes of prokaryotes differ from the ribosomes of eukaryotes. And so they usually say DNA is the third thing that all cells have. DNA, cytoplasm, and cell membrane or plasma membrane. The glycocalyx, also pronounced glycocalyx, is a structure that the cell makes by secreting substances outside the cell. Many prokaryotes can have a glycocalyx. Not all do. In fact, I think the majority of bacteria, as well as prokaryotes, do not have a glycocalyx. It is outside the cell wall, so we say it is outside the cell. The glycocalyx is usually sticky, and there are three types of glycocalyxes. There is a capsule. That's when the glycocalyx is neatly organized. You can see the capsule in the light microscope. The capsule can prevent phagocytosis of the bacteria by a white blood cell. That's the first type of a glycocalyx. The second is a slime layer. The third is the extracellular polysaccharide, or EPS. The slime layer is also secreted by the cell, but it is mostly water. It is loose and unorganized and is not attached very well to the cell. You cannot see the slime layer in the light microscope. Even with the capsule stain, you cannot see the slime layer. It is because the slime layer is mostly water, and therefore it's transparent. You cannot see it in the light microscope. You can see the slime layer if you look in the electron microscope. All right, the extracellular polysaccharide is a glycocalyx that you see in bacteria that are in a biofilm. In fact, the cells secrete the extracellular polysaccharide from the cell, and it's that extra polysaccharide that actually will form the biofilm. Biofilms is a topic we hardly talk about in this class. It's when there's many bacteria growing in a film, and this can put the cells in different environments, like a cell that's near the surface of the biofilm can be richly supplied by nutrients from its host. It has lots of oxygen, and this cell will be metabolically active as well as growing and reproducing, which really means the same thing. And then there can be cells deep in the biofilm, which are essentially in stasis because there's very few nutrients. And that just points out that the biofilm can have different environments, and that can affect the treatment of a bacterial infection that is producing a biofilm. If the bacteria produce a biofilm, it'll be much harder to treat the bacteria than if the bacteria infection does not have a biofilm. The flagella is a structure seen in some bacteria. If the bacteria can swim in a liquid media, or the bacteria can move, it is because the bacteria have at least one flagellum or maybe several flagella. So flagellum is singular, flagella is plural. The flagella is mostly outside the cell wall, this structure here, and this beats to propel the cell in this direction. I'm sorry? Was there a question? All right. The flagella is mostly outside the cell wall, but there is a little bit of the flagella inside the cell wall and a tiny bit of the flagella actually inside the plasma membrane. Flagella is made of chains of protein that we call flagellin. Any questions about the flagella? Remember, if the bacteria can swim or move, it is because the bacteria contain flagella. This is the only way the bacteria can move. At least swim, I'll word it that way. There are 4 basic arrangements of bacterial flagella. You do not need to know this arrangement for any quiz or anything like that, but you do need to answer a lab question. So let me state that there are 4 different flagella arrangements. You can have a flagella on one side of the cell, just one flagella outside the edge of the cell, and that we call monotrichous. Let me blow this up. You can have um, one flagella on both sides of the cell, and this is amphitrichous. You can have multiple flagella on both sides of the cell, and this is lophotrichous. And then you can have flagella coming out all over the cell. So they have flagella everywhere on the cell, on the edges, in the middle, and there's many flagella, and this we call peritrichous. If cells rotate or they run or tumble, it's because the cell has a flagella. And the same thing with swimming. It's because they have a flagella. It's curious when you're watching the bacteria why some of them tend to swim, sort of like a tadpole, and others tumble. I'm not sure what you call that. We call a like a bowling pin, which is tumbling down the bowling lane or something like that. Taxis is the movement toward or away from a stimuli. Bacteria can only move if they have at least one flagellum or multiple flagella. There's two types of taxis: chemotaxis, where the cells respond to a chemical stimuli and then either move toward that stimuli or move away from that stimuli. Here we're looking at phototaxis, and this is when the cell responds to light and the cell either moves towards the light or away from the light. The light source is up here, the cell is moving this way, so these cells are moving away from the light. It's obvious these cells are not photosynthetic cells, because otherwise they'd move towards the light. Any question about any of this? All right, in the spirochetes, we have a special flagella, or actually flagellum, because there's only one, and it causes a rotation, causing the cell to move like a corkscrew moving through cork. Let me blow this up. Here we're looking at the flagella in a spirillum, and this we call an endoflagella or an axial filament, where the flagellum winds around the cell like this. And here we see an artist's rendition of it. The flagellum wraps around the cell this way. One end is anchored at the end of the cell. The endoflagella is inside the cell wall. Let me blow it up one more time. So right there, the light pink is the cell wall, and you can see the axial filaments, or I use the term endoflagella, are actually inside the cell wall, right there. And yeah, the cell wall has a funny structure where the endoflagella is, but it's right there. I'm trying to get that load. Not sure if I'm getting it. Endoflagella cause the spirochetes to move in a characteristic movement. And hopefully we can get this here if there's no commercial.
So last semester, I actually scored 95 for one of my essays in university, and that's crazy. So
There we go. So here's this spirocate, and you see how it just changes its direction by rotating the other way. Let me blow this up. And it can move this way just by moving that way. And there you see how flexible that spirocate was. It was moving up and down this way, and then it bent and then began moving this way and that way. The important part is this is moving in a corkscrew type movement. It's thought that this corkscrew type movement can allow some human pathogens to actually burrow through the mucous membrane. Let me see if I've got this blown up. No, it looks good. Fimbriae are shorter and straighter than flagella, and they are not used for movement. They're used for attaching the cell to a surface. Some human pathogens have fimbriae, and the fimbriae attach to, well, human tissue or human cells. The pili is a structure on a few bacteria that differs from the fimbriae in the sense that there's multiple fimbriae. There's only one pili. And the pili is something for moving DNA from this cell into that cell. So this is usually called the sex pilus. It is longer than fimbriae, and it's used to transfer DNA from this cell into that cell. The pilus, we'll actually talk about it later, it literally will skewer this cell and then can act as a winch to pull the two cells together. The two cells do not need to be together, though, because the sex pilus is hollow, and the plasmid DNA from this cell can leave this cell and then move into that cell. Any question about any of that? All right, let's talk about the prokaryote cell wall. It's a complex semi-rigid structure responsible for giving the cell its shape. The reason why a cell has a cockeye appearance is because the cell wall takes on the cockeye shape, and then it gives that cell that shape. What else did I want to talk about then? Cell wall. The cell wall protects the cell and prevents osmotic lysis. For example, if this cell had its cell wall intact, and then this cell were to fall into a bucket of water, water would then move into the cell, but only up until the cell membrane comes up against the cell wall. Once that happens, the cell membrane cannot expand any further. It's limited by the cell wall. And when that happens, there'd be no room for water to move into the cell, so that the water stops moving into the cell, even though there's higher water concentration outside the cell than inside the cell. Because of this, the cell wall will prevent the bacteria cell from lysing. if the bacteria had no cell wall, or if it were to have a damaged cell wall and were to fall into a bucket of water, water would move into the cell until the concentration of water inside the cell versus outside the cell were to equalize. And probably that wouldn't happen if the cell doesn't have a cell wall. More and more water will enter the cell, expanding the cell greater and greater until eventually the cell membrane pops. And then the cytoplasm will leak out of the cell, and that would kill the cell. The cell wall is located outside the cell membrane, but yet we do consider the cell wall to be inside the cell, as well as part of the cell. I'm not sure why, but they made the cell wall, anything outside of it is considered to be outside the cell. Anything inside the cell wall, including the cell wall itself, is considered to be a part of the cell. The cell wall can contribute to the pathogenicity of some species, like Mycobacterium. Part of the reason why that family is so toxic to humans and other animals is because of the properties in the cell wall that make the cell more pathogenic. And it really depends on what species you're talking about for what this is, but with the Mycobacterium, it would be the mycolic acid makes the cell more pathogenic than if the cell did not have mycolic acid. In most bacteria, the peptidoglycan makes up the cell wall. In gram-positive cells, there's very little inside the cell wall other than peptidoglycan. We will talk briefly about some other molecules in the cell wall. I'll make sure I've got that. Yeah, I did. Peptidoglycan is made up of two saccharides, NAG and NAM. So it's a disaccharide that just repeats, NAG and NAM, NAG and NAM. NAG stands for N-acetylglucosamine, NAM for, oops, way too big, NAM for N-muramic acid, or acetylmuramic acid, sorry. N stands for acetylmuramic acid. And these names probably mean as much to you as NAG and NAM, so I don't care how you learn it. But peptidoglycan is made up of peptidoglycan hooked together one after the other in a row. There will be multiple rows to the side of this row, and then a row above on the plane and below in the plane. Peptidoglycan does have some peptides which help link the peptidoglycan together. And as you can see, this peptide here is linked to that peptide here, which is linked to that peptide there. So this peptide holds this row of peptidoglycan together with this row of peptidoglycan. And then this peptide links on to this peptide and that peptide, these peptides holding the two rows together. This peptide together with these two peptides hold this row to that row. Penicillin actually inhibits the lattice formation of peptidoglycan. And if penicillin gets in here, you cannot have the peptide getting in there and then linking this row of peptidoglycan to that row. And that would cause a hole in the cell membrane, and the cytoplasm can protrude out of this hole. And usually what happens is more and more water enters the cell, and the cytoplasm protrusion gets bigger and bigger as the cell membrane expands and expands. This will happen until the cell takes in so much water that the cell membrane will pop, and that will leak out the cytoplasm out of the cell, which would quickly die. And then sorry, I lost my train of thought. I was talking about peptidoglycan and how it protects the cell membrane, and it prevents the cell membrane from expanding further and further. And so that will prevent lysis of the cell. So the cell wall protects the cell from osmotic lysis. Any question about any of that? All right, here's the gram-positive cell. You can note it has a very thick peptidoglycan layer on top of the cell membrane. There's the cell membrane there. Let me blow this up. That's a little better. Let me blow it up again. There's the cell membrane there. There's the peptidoglycan hooked together with the peptides. We do have a few other molecules in the cell wall, such as teichoic acid and lipoteichoic acid. It's thought that teichoic acid helps hold the peptidoglycan together, meaning this teichoic acid runs from one layer of peptidoglycan to the next, to the next. The lipoteichoic acid does the same. It also has a lipid portion added to the teichoic acid. The lipid portion gets in the cell membrane and then anchors the lipoteichoic acid to the cell membrane. By anchoring it, you give strength to the cell, as well as you I had something else to talk about with DNA, and now I've lost it. I'll try and think about what the heck I was talking about. The teichoic acid, as well as the lipoteichoic acid, is also thought to aid in anions, positive ions. No, that's a cation. Sorry, cations. It's thought to help the cation move through the peptidoglycan. The cation does not require the teichoic acid to help it move through the cell wall. It just tends to do it, meaning the cation will be in solution of water, or in the solution of the cytoplasm if you're inside the cell. And the water can move right through the peptidoglycan. So the cell wall will not hinder the movement of water. And anything dissolved in the water can get through the cell wall, the peptidoglycan cell wall. Any question about any of that? All right. So the lipid portion of the teichoic acid anchors in the cell membrane. The teichoic acid portion of the lipoteichoic acid, as well as teichoic acid, runs through the peptidoglycan, helping hold the layers together. I need to talk. Oh, I already talked about that, so we'll skip it. The polysaccharides and the gram-positive cell wall can provide antigenic variation, which can be used to classify the bacteria. Like, what's this type of, I'll try and look that up and give it to you next time. There's a type of uh, antigenic variation. Trying to remember some names of antigenic variation, and I'm just drawing a blank here, so let me think a minute. Where cells can differ in their antigens so that you can recognize them, and I'm trying to remember some types, like salmonella has different antigenic variation, and depending on which salmonella species variety you have, it can be more pathogenic. One problem with antigenic variation is that if the bacteria have different antigens, meaning different molecules, it's hard to make a vaccine that would affect all the different antigenic variants within the species. All right, let's, oops, I didn't want to be here. Let's get out of there. I think I'm going to end it here, and we will talk about the gram-negative cell wall next time. I really only talked about the gram-positive cell wall. All right, any questions before we go? This is Tuesday, Thursday. All right, if there's no questions, I'll see you next time, unless you're going to be in the lab tonight. I'll see you at 7 in the lab.
Thank you.
All right, bye.